Welcome to the Deep Dive, where we take complex, cutting-edge research and distill it into knowledge you can immediately use. Today, we are going deep on longevity science.
But we're really sidestepping the usual suspects.
Exactly. No rapamycin, no NAD boosters, none of the high-tech genetic stuff. We're focusing on something that is, fundamental. It's already in your body. We're talking about collagen.
And it's a complete pivot in perspective. For decades, collagen has been seen as, well, a structural aid, a bulk supplement for better skin, maybe healthier joints.
Drum your nails.
Right. But the sources we're digging into today, especially this really fascinating, dense study in NPJ aging, they suggest we have fundamentally underestimated this protein and its simplest components.
Okay, so let's unpack that shift. We're moving beyond thinking of collagen as just like raw material. The new frontier is this idea that the absolute minimum building block of collagen, just 3 amino acids in a very precise ratio, it doesn't just provide material, it acts as a powerful biological signaling molecule.
A conserved one. It triggers these profound pro-longevity pathways across totally different species. It's a truly minimalist recipe for extending health span.
So our mission today is to follow that journey. We're going to trace this signal from, simple lab worms all the way to human cells.
All the way to an observational trial, yes, where people took a cocktail based on this recipe and saw a significant reduction in their biological age. We need to get into the rigorous science behind this 3.1.1 secret.
And these sources are dense. They have to be because the claim is so big. This NPJ aging study is really a masterclass. It tracks this amino acid mixture from nematodes to human cells to old mice and then to people.
And that complexity is necessary. They're trying to prove that this response, this mechanism, is ancient, that it's been conserved across the animal kingdom.
And the big reveal, the anchor for this whole deep dive, is this central finding.
It is. Researchers figured out that a precise 3-to-1-to-1 ratio of 3 very common amino acids, glycine, proline, and hydroxyproline, is enough all by itself to trigger these massive pro-longevity effects in model organisms.
It's like our biology is just exquisitely tuned to recognize this minimal collagen signature as a cue to, start maintenance and repair.
That's exactly it. It's a signal to get to work.
So before we get to that specific ratio, we really have to grasp the sheer scale of collagen's importance in our bodies. I mean, this isn't just a vanity protein. It's the ultimate scaffolding.
It absolutely is. It's not just abundant, it is dominant. If you look at your total body protein, collagen makes up something like 12% in females and even higher, about 17% in males.
Wow.
It's woven into literally every single load-bearing tissue you have. Skin, bone, cartilage, tendons, ligaments, your entire vascular system. So losing collagen is fundamentally losing your structural integrity.
And the decline is relentless. It's one of those visible signs of aging.
Without a doubt. Just think about skin aging. Your skin's collagen production decreases by approximately 1 to 1.5%. Every single year.
That sounds small, but it adds up.
It compounds decade after decade. And that loss is the primary driver behind fine lines, deeper wrinkles, reduced elasticity, and a measurable drop in hydration. The structure just breaks down.
So we know we need it, and people supplement with it, but how does it actually get into our system? I think there's this misconception that the powder you drink just travels intact to your skin or your joints.
That's a really crucial point. Ingested collagen, even the hydrolyzed peptides, is still way too large to be absorbed whole. It has to be broken down.
By stomach acid?
By gastric enzymes, mainly,
Yeah.
into these much smaller units called di- and tripeptides. So that's just chains of 2 or 3 amino acids linked together.
So the key is the big protein. It's these tiny molecular signals that survive the breakdown process. How do they get from the gut into the bloodstream?
They rely on this highly specialized piece of machinery in your small intestine. These little peptides get absorbed into the gut cells, the enterocytes, through a specific transporter protein. It's called PPT1, peptide transporter 1. And it's critical because it's super efficient at grabbing these short peptide chains. It allows for a much higher uptake rate than if your body just had to absorb individual amino acids one by one.
So once that little peptide is inside the gut cell, what happens?
That's where the final processing happens. Most of them are broken down even further by enzymes inside the cell into individual amino acids. Then they're finally released into the bloodstream, ready to be used by tissues all over the body.
So the efficiency varies a bit.
It does. Absorption rates are typically cited somewhere between, say, 39% and 60%. But this whole process breakdown, PEPT1 transport final breakdown, that's why the researchers realized the longevity signal had to be in those small peptides or in the specific profile of the amino acids they release.
And even before this new discovery, we already had a ton of clinical evidence showing that general collagen supplementation provides real benefits, right?
Oh, absolutely. The benefits are numerous and well established. In dermatology, for example, you have multiple placebo-controlled trials showing that supplementing for about 12 weeks measurably improves skin hydration, it boosts elasticity, and can reduce fine lines and even cellulite.
And what about deeper than the skin? Our joints, our bones.
That's where it really gets into true physical health span. We see improvements in brittle nails. And for bones and tendons, which are so collagen dependent, you see significant structural
Like
improvements.
making them stronger.
Exactly. Intake has been shown to increase the cross-sectional area of the patellar tendon. For bone health, it increases bone mineral density, which is huge for populations at risk of bone loss.
And for active adults, what about pain and injury?
That's a major area. For young active adults, oral collagen has been shown to reduce activity-related joint pain, especially in the knee. And for older folks with osteoarthritis, consistent intake over 4 to 6 months can decrease pain and improve their ability to be physically active.
What's really interesting is that the benefits go way beyond connective tissue. It hints at this metabolic signaling role that we're about to dive into.
Yes. Think about sarcopenia, the age-related loss of muscle. You combine resistance training with collagen, and you get this additive benefit. You see an even greater increase in lean body mass and hand grip strength.
And it's not purely dependent on the exercise, which suggests an independent signal.
That is the key takeaway. Even in young, sedentary men, just taking collagen peptides was shown to increase the synthesis rate of muscle connective protein.
So it's not just passive building block.
It's a trophic signal. It's a nutrient cue that tells tissues to build new protein. And metabolically, in type 2 diabetic patients, studies showed significant improvements across the board. Lower fasting glucose, lower HbA1c, better cholesterol, and improved insulin sensitivity. It suggests a really systemic benefit.
So we have this universally important protein. It declines with age, and supplementing with its fragments delivers these broad benefits. The obvious next question is, if collagen is so complex, you said 28 types in humans,
And 184 in the C. elegans worm.
so which specific part of all that is the universal longevity signal the body is actually looking for?
And that was the brilliant question the researchers asked. They decided to look for the minimalist recipe.
So they needed to move past the idea of just bulk protein and isolate this core signaling component.
Exactly. They knew that general collagen supplementation could increase the health span and lifespan of model organisms, but they needed to find that minimal required unit, the thing that actually triggered the anti-aging response.
Okay, so let's go back to Collagen Chemistry 101. What's the foundational structure that dictates this ratio we keep mentioning?
The structure of collagen's triple helix is defined by its repeating sequence. It's always glycine XYN.
Okay.
The X position is usually proline, and the Y position is often hydroxyproline. But the absolute critical constraint is that glycine, because it's the smallest amino acid, has to be at every third position. It's the only way the three helical strands can pack together tightly enough to be stable.
Ah, so that structural rule is what creates the ratio.
Precisely. This biological mandate means that glycine content is naturally 3 times higher than the others. That's the origin of the 3.1.1 ratio. The researchers hypothesized that this minimal signature, the structural echo, would be the longevity cue.
And they tested this on the nematode worm, C. elegans. How do they actually track the longevity signal inside the worm?
They used a surrogate marker for aging. It's a reporter called PACOL144.GFP. It basically just tracks the expression of the worm's own collagen genes.
And that declines with age, just like in us.
Yep. It starts to decline around 6 to 8 days old. So the researchers wanted to see if their 3.1.1 mixture could keep that collagen engine running for longer.
And first, they had to prove it wasn't just one powerful amino acid doing all the work.
Yes, that was critical for proving synergy. They fed the worms glycine alone, proline alone, or hydroxyproline alone, and the finding was definitive.
Nothing happened.
Nothing. None of the individual amino acids did anything to stop the age-related decline of collagen expression. They provided raw material, but the signal for maintenance was completely absent.
But when they combined them,
The synergy was just undeniable. When they put glycine, proline, and hydroxyproline together in that precise 3.1.1 ratio, they saw a dramatic, highly significant prolongation of collagen expression during aging. The cells clearly recognized that specific signature and fired up their internal repair protocols.
and this molecular response translated directly into more time on the clock.
Yeah.
What were the lifespan results?
The results were very compelling. Across 5 separate trials, supplementing with the 3.1.1 ratio significantly increased the mean lifespan of the worms by a range of 6% to as much as 27%.
How does that stack up against just giving them bulk collagen?
That was a vital head-to-head comparison they did. They used bulk rat tail collagen. And while the bulk protein did increase the maximum lifespan a little, it only mildly affected the mean lifespan.
Meaning most worms didn't really benefit.
Right. But the isolated 3.1.1 mix, it increased both the mean and the maximum lifespan more robustly. It suggests the functional longevity signal is really concentrated in that minimal ratio. It actually outperformed the bulk protein.
And they tested how sensitive this signal was. Did the ratio have to be perfect?
They did a lot of testing on that. They found that if you increase the concentration of any of the 3 amino acids, you'd get a further increase in lifespan. So they were all clearly core components.
But the importance of the combined signature, that held up.
It did. They tested skewed ratios, like 1 glycine to 3 proline to 1 hydroxyproline. And they found that all these combined ratios still gave similar strong longevity benefits. So it's the combined presence of all 3 in that structural stoichiometry that triggers the response. It's a synergistic effect, not just simple addition.
And the final proof that you need all 3.
That was the hydroxyproline emission test. They took it out entirely, a 3 glycine to 1 proline to 0 hydroxyproline ratio, and the lifespan increase was noticeably smaller than with the full 3.1.1 ratio. It really emphasized that the unique benefit requires the complete combination.
So they didn't just live longer, they lived better. What about health span?
They measured vitality using thrashing rates in liquid, which is a proxy for movement and activity. And the 3.1.1 ratio consistently improved those health span metrics. In fact, the worms fed this ratio were significantly more active than the controls at a very old age, over 24 days. The maintenance signal clearly extended their period of youthful function.
Okay, so we've established that the minimal core components of collagen in their natural ratio are recognized by worms as this profound signal to repair and extend vitality. Now we have to see if this mechanism is actually relevant to us, to human biology.
Right, that was the next major step. They had to confirm this wasn't just some quirk of nematode metabolism. They needed to see if that same 3.1.1 signal triggered the same maintenance response in human cells.
And for that, they used human dermal fibroblasts, the main cells that build the matrix in our skin. What was the setup?
They treated these HDS with the exact 3.1.1 ratio, 3 millimolar glycine, 1 millimolar proline, 1 millimolar hydroxyproline. And then they did RNA sequencing to see which genes got turned on or off.
And what was the key molecular instruction the cells received?
It was a robust and immediate signature. Within just 8 to 24 hours, the cells showed a highly significant upregulation of genes related specifically to collagen and the extracellular matrix.
Wow.
Yeah, the analysis highlighted categories like extracellular matrix, structural constituent, and collagen-containing extracellular matrix.
So that's powerful confirmation. The human cells recognized the 3.1.1 ratio just like the worms did, and they responded by fire up their own structural repair programs.
It strongly suggests that this ratio acts as a universal ancient recognition signal. It tells cells to ramp up their own production of collagen and remodel their environment.
And beyond just the structural genes, what else was happening inside the cells metabolically?
That was really interesting. While they were ramping up collagen production, the treatment also caused a noticeable downregulation of ribosomal components.
The factories that make general proteins.
Exactly. And a less robust, but still present, downregulation of mitochondrial pathways.
That shift in resource allocation is fascinating. It's like the cells are prioritizing quality maintenance over just general growth.
That's a great way to put it. It's not a growth signal, it's a specialized maintenance signal. It's shifting resources toward long-term structural integrity, which mimics a metabolic state you often see in other longevity interventions.
So now the researchers decided to maximize the effect for the human trial. They moved from this simple amino acid blend to what they called a collagen activator formulation. What was the primary addition?
Alpha-ketoglutarate, or AKG, is a very popular longevity compound in its own right, but its role here is very specific and structural.
How does AKG help build collagen?
AKG is the critical rate-limiting cofactor for an enzyme called prolyl hydroxylase. This enzyme is what adds a hydroxyl group to proline, turning it into hydroxyproline. That step is absolutely vital for making a stable collagen triple helix. So by adding AKG, they were basically providing the fuel for the cell's construction machinery.
So they've got the raw materials, the 3.1.1, and the tool fuel, the AKG. But AKG is known to extend lifespan on its own. So how did they prove the 3.1 point ratio was still doing something independently?
They used a really brilliant, rigorous test back in the worms, and they showed the benefits were additive. This suggests they work through parallel pathways. AKG increased lifespan on its own, and when you combined it with 3.1.1, you got a stacked, even greater effect.
And they had molecular proof of these distinct pathways.
Yes, using a specific mutant worm strain, the DPI-18 mutant. This strain has a broken prolyl hydroxylase enzyme, the one that AKG needs to work. And in this mutant, the longevity effect from AKG was completely gone. It couldn't work. But critically, the 3.1 amino acid supplementation still worked in these same mutants.
That's the elegant proof. So the 3.1.1 is a direct signal, independent of AKG, And the two benefits just stack on top of each other.
Exactly. The final ingredients in the cocktail were vitamin C, which is essential for collagen synthesis, and 4 milligrams of astaxanthin.
A really potent antioxidant.
Precisely. Astaxanthin is known to improve collagen homeostasis and has shown lifespan extension in a bunch of models. So that was the final collagen activator formulation.
And with that final formulation, they tested its safety and function in old mice.
Right. 20-month-old male mice, which is roughly geriatric. They gave them the supplement in their diet for 6 months.
And the first test is always safety.
It was very well tolerated. The dose was modest, only changing total protein intake by about 5%. Body weights, organ weights, all stayed pretty similar to the controls.
Okay, but let's address a critical limitation here for our listeners. The study lacked what are called isonitrogenous controls. What are those, and why does their absence matter?
So, isonitrogenous controls mean that the control diet has to be perfectly matched to the intervention diet for total nitrogen or total protein.
So they would add a dummy protein to the control group's food.
Exactly, something non-functional like casein, just to match the protein level. Since the intervention group got about 5% more protein from the 3.1.1 ratio, without that control, we can't completely rule out that some of the benefits were due to just a modest general increase in amino acids rather than purely the specific 3.1.1 signal. It's a necessary scientific caveat.
Got it. But despite that caveat, what were the two major functional benefits they saw in these old mice?
Two really relevant anti-aging effects. First, strength. The supplemented mice maintained their maximum grip strength over the whole 6 months. This is huge because the control group showed the expected age-related decline.
And the second one.
Body composition. The mice had significantly less visceral fat accumulation, specifically the epididymal white adipose tissue, compared to the controls.
And targeting visceral fat in an aged model is a massive marker for metabolic health, right?
It is paramount. Visceral fat is highly inflammatory, and it's strongly linked to age-related conditions like metabolic syndrome. So reducing that suggests the cocktail induced a really favorable metabolic shift, helping the mice keep a more youthful body composition.
That is a very strong package of preclinical data. A conserved signal in worms, molecular activation in human cells, and maintained strength and less fat in old mice. This takes us right to the human trial.
Right, the human arm. This was an open-label, single-arm observational trial. And it's crucial to understand what that means. There was no placebo group. Everyone, participants and researchers, knew what was being taken.
So we have to reinforce the caution here. We're looking at a cocktail 3.1.1 plus AKG, vitamin C, astaxanthin in an uncontrolled setting. How should we interpret these results?
The results are excellent for generating a strong hypothesis. They validate the safety and functionality, but they are not definitive proof of rejuvenation. We can't rule out placebo effects, selection bias, or the known benefits of the AKG component. We have to see it as evidence that the cocktail works, but we need randomized trials to isolate the 3.1.1 effect.
Understood. So what was the intervention and who are the participants?
They took one daily sachet with 8.4 grams of the 3.1.1 mix, 1 gram of calcium AKG, plus the astaxanthin and vitamin C. And the cohort was unusual. The mean age was about 47, and they were overwhelmingly healthy, well-educated, highly active, and already taking other supplements.
A very healthy baseline, which makes any improvement pretty compelling, but also harder to generalize. Let's start with the skin improvements they saw at 3 months.
The skin benefits were significant and they were rapid. Cheek hydration, measured objectively, started at a clinically very dry level. By the end, it had increased dramatically into a sufficiently moistened clinical state. This points to real systemic structural improvement.
What about elasticity?
Elasticity also significantly improved, which suggests the signaling was promoting repair in the skin's deeper layers. Wrinkle severity trended downwards, but that change wasn't statistically significant over just 3 months.
Okay, but the headline finding was the reduction in biological age after 6 months. How did they measure that?
They use the True Age Epigenetic Methylation Saliva Clock. It's a powerful tool based on epigenetics, how your gene expression changes over time. These clocks analyze methylation patterns, which are chemical tags on your DNA that change predictably with age.
Can you break down what a CPG site is? They looked at 9 of them.
Sure, a CPG site is just a specific location on the DNA strand where a cytosine nucleotide is followed by a guanine nucleotide. Methylation at these sites acts like a dimmer switch for nearby genes. The True Age clock focuses on just 9 specific CPG sites whose combined methylation status correlates extremely well with chronological age and, more importantly, can predict health status and mortality even better than your birthday.
So after 6 months of taking this cocktail, what did the clock say?
Well, while the participants' chronological age went up by half a year, their mean biological age decreased by an average of 1.37 years.
Wow.
It was a statistically significant reduction. They effectively aged backward by almost a year and a half in just 6 months, and it was very consistent between men and women.
What about those staggering figures for the high responders? I saw reductions of up to 12 years. What's the statistical explanation for that?
Those maximums are dramatic, but we have to talk about regression to the mean. These clocks have some inherent measurement variability. So, a participant who starts with a biological age that's much older than their real age, maybe due to a temporary bit of inflammation when they took the first test, is statistically likely to score closer to the average on the retest.
So the intervention likely helped, but those extreme numbers are probably a mix of the supplement and just statistical drift.
Yeah.
The 1.4 years is the reliable figure.
Exactly. That's the one to focus on.
Did they find any lifestyle factor that interfered with the supplement's effectiveness?
They did, and it was a crucial one. Individuals who consumed more than one alcoholic drink per week showed significantly fewer improvements in their biological age compared to non-drinkers. It suggests the metabolic stress from regular alcohol consumption might be fighting against the benefits of the cocktail.
Okay, so we've established the 3.1.1 ratio is a powerful signal, but the human intervention was a cocktail. To really do a deep dive, let's go back and look at the known longevity mechanisms of the 3 core amino acids themselves.
Absolutely. Let's start with glycine, the most abundant one. Glycine is heavily involved in nutrient sensing.
How did it interact with the methionine cycle?
Glycine's longevity benefits come largely through the methionine cycle and one-carbon metabolism. Methionine restriction is one of the most reliable ways to extend lifespan in animals. Glycine helps the body conserve its own reserves of other amino acids needed to regenerate methionine.
So by providing extra glycine, you're sort of mimicking some of the benefits of methionine restriction.
That's a great way to put it. You get a lower growth signal and better metabolic flexibility without having to cut your protein intake. It's also profoundly anti-inflammatory, neuroprotective, and has been shown to improve sleep and cognition in humans.
What about proline? And what's the significance of its pathway not involving GCN2?
Proline is tied to mitochondrial function. In senescent human cells, proline can help restore it. In the worms, its longevity effect depends on mitochondrial complex 2. The critical finding, as you mentioned, is that its effect does not depend on the GCN2 amino acid sensor.
And GCN2 is the universal, we're out of protein sensor.
Right. The fact that GCN2 wasn't involved tells us that proline is acting as a specific metabolic signal, not just tricking the cell into thinking it's starving for protein in general.
And finally, hydroxyproline, the one that ensures synergy.
Hydroxyproline is unique because, as a free amino acid, we can't actually reincorporate it directly into new collagen. It mostly gets metabolized into glycine, but its presence completes the signal, and it has its own benefits. It's been shown to promote muscle growth in some models and has anti-inflammatory effects in others.
So we have this cocktail of three metabolic actors that are also structural components plus AKG. Let's contextualize that 1.4-year reduction in biological age. How does that stack up against other interventions?
It's a strong result. It's comparable to other targeted nutritional studies. For example, vitamin D supplementation has been shown to reduce biological age by about 1.9 years in certain groups. Even comprehensive lifestyle changes have shown about a 2-year reduction.
And how does the likely contribution of AKG factor in?
The Rejuvenation study, though it was also observational, reported an incredible 8-year average reduction over 7 months using an AKG formulation. So AKG is clearly very powerful.
Which makes this result even more interesting.
It does. The fact that the collagen activator got a strong 1.4-year reduction with a relatively low dose of AKG, just 1 gram, suggests the 3.1.1 ratio is contributing significantly to the overall effect.
This brings us to the final critical analysis. The preclinical science seems impeccable, but the human data is really more of a hypothesis at this stage.
That is the essential message. The molecular effects are conserved across species, suggesting a really ancient, powerful mechanism. But the human findings are subject to those 3 main caveats. It was open label, it was a cocktail, so we can't attribute the benefit just to the 3.1.1, and the mouse study lacked those isonitrogenous controls.
So we need randomized placebo-controlled trials to really isolate the contribution of the 3.1.1 ratio versus the AKG.
Precisely. This study is an incredibly promising foundation. It just urges more investigation into this minimalist approach to tissue health.
So finally, let's connect this strategy to the broader societal implications of increasing longevity. If we can reliably extend health span, what does that mean for how we all live?
Oh, it changes everything. The need to invest in health span, the years you live without chronic disease, becomes critical. If you can maintain youthful function, you reduce the burden on healthcare systems, and you enable much more productive aging.
And this feeds into models like what we see in the blue zones.
Yes, blue zones work because they facilitate low-stress, active, communal lives. If we use nutritional science to maintain the body's physical integrity, strong muscles, mobile joints, healthy metabolism, we just increase the window during which people can actively participate in those kinds of lifestyle.
And the financial implications.
They're profound. Organizations are analyzing how longevity increases the risk of, running out of savings. If an intervention like this can delay the onset of expensive chronic diseases by 5 or 10 years, that has immense positive consequences for financial security and the global economy.
And for the workforce, it means longer, healthier careers.
If people are biologically younger, they'll choose or need to work longer. So society has to adapt. We need to ensure digital literacy and continuous reskilling for older workers. An intervention that promotes collagen health isn't just about skin. It's about maintaining the vitality you need to be a productive member of society for longer.
So if we synthesize the core takeaway from this entire deep dive, it's that this simple collagen-mimicking 3.1.1 ratio is not just building material.
It appears to be a crucial, evolutionarily conserved signal for initiating structural repair and ECM homeostasis across the entire biological spectrum.
It really reveals the power of substrates. I mean, this shows that sometimes the most potent path to longevity isn't a complex drug. It's about giving the body the exact fundamental building blocks it needs.
And combining them with the right enzymatic support like AKG and vitamin C to optimize the cell's own internal maintenance machinery.
It shifts the focus from treating symptoms to just supporting the body's own ancient ability to repair itself.
It's a very elegant, minimalist approach to geroscience.
So here's the final provocative thought to leave you with. The reduction in biological age in the human trial was significantly associated with objective improvements in skin thickness and hydration.
Mm-hmm.
If these visible structural skin changes and these core epigenetic biological age markers are so tightly linked, how deeply might this simple 3.1.1 signal influence the aging of deeper, unseen, collagen-rich organs like the heart, the liver, your blood vessels, the structural integrity of your brain?
It leaves us with a powerful question. Is optimizing our most abundant structural protein by providing its specific, minimal signature one of the simplest yet most systemically powerful strategies for optimizing our entire body's lifespan?
Something profound to consider until our next deep dive. We'll see you then.